Hello, everybody. It's Mr. Caudell. We are here in the middle of Chapter 11. Specifically, we are at 11.3 and 11.4. We've learned about the signal transduction pathways, and now we just need to finish. We've learned a lot of vague stuff, and we're going to get to some specifics on the last two sections of it, which are transduction and then response. I thought I'd start off with a movie. Okay, thanks. Couldn't you just watch it all day? What does that have to do with signal transduction pathways? I have no idea. I think it's just something we all needed to see some greatness. Are you feeling great right now? Probably not either. I'm just kidding. Eventually, aspire to be great. Do you think that kid told his parents when he was growing up, I'm going to be the best tennis ball juggler in the world? Maybe he did and maybe he didn't, but the point is it doesn't matter what you do in your life. What matters is that you're all in and that you do it to the best of your ability. Work harder than anyone else. Be great. Don't be mediocre. And it could be a tennis ball juggling. It could be anything. Just find your passion. Not now. You may already know it, but most kids don't. Just figure it out over the next 4 or 5 or 6 years, and maybe later than that it will come to you, but then outwork everyone else. Be the best tennis ball juggler. I'm just kidding. In the world. Let's finish this. I'm going to tell you this is probably the hardest stuff that you've seen in this class so far. Also, there are definitely pages in these two sections of 11.3 and 11.4 that I do not want you to read. So make sure that you go where I've described what is important and look at that before you begin reading. Well, I've left up the term block day here. I hope that it still applies to your year. I love block days. And we have an enzyme lab that we do every year. It's part of the AP Biology curriculum. Hopefully we're going to do it next block day, but I don't know what class is looking like these days. It's on catalysis, breaking down stuff using enzymes, essentially. Well, remember that there are 3 stages of cell signaling. There's the reception of the ligand or the signaling molecule that then changes the shape of the receptor, which causes this cascade of stuff that we call transduction. The message is being transduced, maybe changed a little bit into different forms, and then eventually there's going to be some sort of response at the end. Usually it says up here an activation of a cellular response. I'm going to tell you most of the time that is something going into the nucleus, like a transcription factor binding onto a certain segment of nucleotides on a gene on DNA, and then essentially probably knocking off, we'll learn about this later, some sort of inhibiting protein, and then allowing transcription to take place. That message is translated, and then a protein is made. So that's usually what's happening there. But let's get to the middle here, transduction. There's a definition. Cascades of molecular interactions relate signals from receptors to target molecules in the cell. Well, that's pretty vague. Let me give you some specifics. Well, if this is the first messenger, this ligand or signaling molecule that is bound to the G protein coupled receptor, And this is the second messenger, the stuff that's now into the cytoplasm there. We kind of saw the G protein coupled receptor cartoon the last time we got together, but we haven't seen it in this much detail yet. We define second messengers. They are small non-protein water-soluble molecules. Okay, so that's many atoms together, or ions. That could just be like a sodium ion or a chloride ion or potassium ion. These are charged atoms that have either lost or gained electrons, and they spread throughout the cell by diffusion, which tells me that they're going to be they're going to be very specific in their targets, but it's going to be non-specific as they're just diffusing out into random space. And then number two, that there's going to be some sort of amplification of this signal. So those are the two major reasons why transduction occurs. Second messengers they participate in pathways initiated by G protein coupled receptors. There's a couple of them that they're going to outline in your book, IP3, which I'm not that interested in, but CAMP, cyclic AMP, I'm super interested in. If you remember this yesterday, this kind of generic cartoon, you'll see the protein that exists in the membrane that's going to be activated by the G protein, which is kind of turned on by GTP phosphorylating it and then becoming GDP. It slides over, and now we have this active enzyme where we once have an inactive one on the top right corner of your picture here. And this now has a name, and I want you to commit this to memory and find this in the textbook. It's called adenylyl cyclase. Okay, so what's the job of adenylyl cyclase? So we know everything that's happening there with those three things in the membrane. And what does it look like the job of adenylyl cyclase is? Well, it says it's an enzyme in the plasma membrane, and it's going to convert ATP to CAMP in response to an extracellular signal. CAMP, or cyclic AMP, is a second messenger. Go ahead and circle that if you have a handout or find it in your textbook. So if we have adenosine triphosphate, and now I'm telling you that gets turned into CAMP, what do you think that is? I'll tell you what the C is. It's cyclic, and then what is AMP? That's monophosphate. So what that suggests to me is that two phosphates are going to be knocked off the ATP here, and it's actually going to be the molecule of ATP is going to be totally changed into adenosine monophosphate, and it's a circle or cyclic somehow. So it's one of the most widely used second messengers, and you can see here, I wish I could point to this, you can see here as it goes from left to right that two phosphate groups indeed get knocked off that thing. And then you can see, just looking to try to point this to you, you can see where it says CAMP and the diamond in the middle there, you can see it's a circle. Do you see how the covalent bonds have been formed there? And there is a kind of a circle, cyclic. Well, eventually this could be, I guess water could be added to this using the enzyme phosphodiesterase, and then we can stop cyclic AMP. And remember, the whole idea here is if cyclic AMP is one of these messengers, it's great, but we need to be able to stop it as well, and it shows you here how it is stopped. Water is added, the kind of cyclic or circular portion of the molecule gets broken, and we just have AMP. And that probably, the adenosine monophosphate is probably going to go back to the mitochondria and grab two more it's going to be phosphorylated two more times through cellular respiration, and then we have our energy storage molecule we use called ATP again. So it'll probably be recycled. Well, eventually, cyclic AMP is going to turn on something called protein kinase A, and I'll tell you what that is in a second. Make sure you got to know this picture inside and out. You should be able to close your eyes before the exam and draw this picture without looking at anything. You got to know this. G protein coupled receptors are the most widely used in the body. This is called PKA for short, protein kinase A. I'll be quiet. Explain what's happening here to your neighbor. Well, it's time to watch two movies on this, and what I want you to do is I want you to be amazed, and I want you to bring your parents in the room and then have them watch the movies with you and say, sounds like a foreign language, doesn't it? Not to me. You should be amazed at how much you can understand while you watch these. Be proud of all that you've learned.
Adenylyl cyclase is a membrane-bound enzyme. whose catalytic domain is activated by the GTP-bound form of the stimulatory G protein alpha subunit, or G-alpha-S for short. Activated adenylyl cyclase converts ATP to cyclic AMP, which then acts as a second messenger that relays the signal from the G protein-coupled receptor to other components in the cell. In most animal cells, cyclic AMP activates cyclic AMP-dependent protein kinase, or pKa. In the inactive state, pKa consists of a complex of 2 catalytic subunits and 2 regulatory subunits. The binding of cyclic AMP to the regulatory subunits alters their conformation and liberates the catalytic subunits, which are now active and phosphorylate specific target proteins. In some endocrine cells, for example, the activated pKa catalytic subunits enter the nucleus, where they phosphorylate a transcription factor called CREB. Phosphorylated CREB then recruits a CREB binding protein. This complex activates transcription after binding to specific regulatory regions that are present in the promoters of appropriate target genes.
One example of extracellular signaling is a liver cell's response to the hormone epinephrine. In times of stress, epinephrine is released into the bloodstream from the adrenal gland. Upon reaching the liver, epinephrine binds to beta-adrenergic receptors on the surface of liver cells. Binding of epinephrine causes a conformational change in the receptor, allowing it to associate with a trimeric GS protein. Binding to the receptor creates a change in the GS-alpha subunit, resulting in GDP displacement and GTP binding. The GS-alpha GTP complex dissociates from the G-beta gamma subunits, then binds and activates adenylate cyclase. Activated adenylate cyclase catalyzes the formation of cyclic AMP from ATP. The effects of cyclic AMP are mediated through cyclic AMP-dependent protein kinases, or CAPKs. Cyclic AMP binds the regulatory subunits of CAPK, which releases and activates the catalytic subunits. A catalytic subunit enters the nucleus and phosphorylates one of its multiple targets, the CREB transcription factor. Phosphorylated CREB binds cyclic AMP response elements, or CRE sequences. With the coactivator P300-CBP, it stimulates transcription of CRE-regulated genes. In liver cells, CRE-containing genes encode enzymes involved in glucose production. The end result is increased levels of glucose, necessary to energize tissues during the stress response.
Pat yourself on the back. Isn't it awesome? I hope I'm not disappointing some of you and you're going, oh my gosh, I'm so lost right now. I hope that you were able to recognize some of that vocabulary and see it in kind of a real world example. I get it, that's a cartoon on a screen, but hopefully that it makes a little bit more sense. Well, next picture is this. This is in the 10th edition. This is on page 219. This is a big deal, this picture here. It's going to show you what I've been simplifying with all these arrows. Once protein kinase A is turned on by this cyclic AMP, you can see here that a lot of steps are going to happen, and they kind of look the same. I'm going to read this. It says, like following dominoes, the receptor activates another protein, which activates another, and so on, until you get some sort of response down at the bottom, until a protein producing the response is activated. We'll get to this detail in just a second. Go ahead and teach your neighbor what you think is going on here. What do you see when you look at page 219 in the 10th edition? Well, let's get into some details. So this term protein kinase, it just refers to an enzyme that phosphorylates proteins. That's what it does. So it grabs a phosphate group off of ATP, creating ADP, or adenosine diphosphate, which goes away, but it takes that phosphate group and it phosphorylates a protein. And often, at least in the transduction portion of signal transduction pathways, when that occurs, that turns on an enzyme that then goes and turns on another enzyme and turns on another enzyme. And you can see that it says, in many pathways, the signal is transmitted by a cascade of protein phosphorylations. So what you see here is that active protein kinase 1 grabs a phosphate group, and then it turns on active protein kinase 2, which then grabs a phosphate group, and turns on active protein kinase 3, which grabs a phosphate, and then it turns on, way down there at the bottom right corner, the active protein turns that on with the phosphate on it now, and then we get a response. So the point is that it's just a bunch of enzymes that are grabbing phosphates from ATP and then phosphorylating other enzymes, turning them on, and you get this, it's called the phosphorylation cascade. This is all these arrows here that we see on the board. Well, you can see here it says protein kinases transfer phosphates from ATP to protein in a process called phosphorylation. You can see this, this protein that was once a circle grabbed a phosphate group off of ADP plus a phosphate, which is ATP, or 3 phosphate groups. And then now it's a square. And the point is it's going to do that again and again. But like I've said, we have to have this under control, because if you can imagine, the creation of a protein is really energetic. It takes a lot of resources from the cell, and it can't go on forever. You have, this is fine. A message came, it said, do this, create this protein, and we're doing it. But we have to be able to shut it off. And so the way that we shut it off and someone told me that these are the little PPs that you see on this picture on page 219 in the 10th edition. These are called protein phosphatases. And you can see there, they're going to strip that phosphate group off of the activated enzyme, basically shutting down the enzyme so it can't continue in its portion of the phosphorylation cascade. And then it's going to rephosphorylate ADP eventually to become ATP. But the point, the key point here is that it takes the phosphates from proteins in a process called dephosphorylation. You can see that again and again and again. It's just us being able to shut down this process at a variety of different points. You can see that on there where it says the little PP things. Just grabs phosphate groups. This phosphorylation and dephosphorylation system acts as a molecular switch, turning activities on and off. So there's a lot of control here. Go ahead, I'll be quiet. Explain this to your stuffed animal or your neighbor, please. Well, let me give you some real world application for some of this. And I think this is fascinating because you'll be able to see terms that we've learned in this chapter applied to structures that you've seen before inside of organisms. So here's a compound called THC. It's the active chemical in marijuana, and it has a receptor that it binds to in the nervous system. So what we're seeing here is the end of it's called an axon terminal. It's at the end of a neuron, and the idea is that if the electrical signal comes to the bottom of this, it's going to release neurotransmitters across the synaptic cleft and then stimulate another neuron. What THC does is, you can see up there in the upper right corner, is that it binds to a receptor. You can see right below it to the right of the cannabinoid CBI receptor is a G protein. Then you can see adenylyl cyclase. Then you can see ATP that's going to be turned into CAMP. Then that's going to turn on a protein kinase, which is going to phosphorylate something. And then all around the outside, you see these ligand-gated ion channels. Essentially, when these things are phosphorylated, their shape changes and a gate opens, and then ion can go through. And then that's going to either turn off or turn on another neuron. And so then, meaning, let's imagine that someone gets marijuana in their system. It's going to cause some sort of response that you associate with marijuana use by either turning on or turning off another neuron. Here's another one, aspirin. It's just a molecule with a bunch of carbons, hydrogens, and oxygens, essentially. And what it does is it's going to bind right here. And it's going to stop this whole cascade in the right side there. You see all those arrows, and basically it's going to stop all those things. That's kind of what that thing looks like, that arrow that's flat there. Another one is how green tea works. And in preparation for this, I kept looking at this and I was like, I gotta trust myself. This has something to do with green tea. Let's just imagine that let's not imagine that it does. Let's look at this big thing. Be overwhelmed by this. This is just a few pathways inside of cells which are stimulated by signal molecules which have bound to receptors and started these signal transduction pathways. There's a ton of acronyms here. You don't need to know how they work. Just you can look for things that have K on them. Probably everywhere you see a K, that's a kinase, which is going to phosphorylate something else in that signal transduction pathway. You can see way down there at the bottom, there's gene regulation in the bottom right corner. I think it's pretty amazing, and I want you to be overwhelmed that this is an oversimplification. This is fine for these 5 or 6 or 7 pathways, but there's probably 1,000 pathways in each cell, and we don't even know how they all work yet. We have students from here that become biochemistry majors all the time in universities, and this is the type of stuff that they're trying to figure out. It's pretty amazing. I hope that you're in awe of it. Well, way down here is the final thing, response. So if we want to activate a cellular response, it's often the creation of a protein following transcription and then translation. And I've said that before. You can see this in this picture. The cellular response is often transcription to create a protein product, and you can see there that something's going to happen. We see that phosphorylation cascade box in the middle that we just went through. Something's going to be phosphorylated, a protein. It's going to go into the nucleus. It's going to activate an inactive transcription factor. What do you think a transcription factor is going to do? It's going to turn on transcription. If it's activated, you can see a phosphate group on there, and now all of a sudden mRNA is made and eventually a protein. It's pretty amazing. Please push pause and explain this to your neighbor. Well, we're almost done with chapter 11. There's just 2 more concepts left. First one is signal amplification. What I want you to appreciate from this picture is that there's one binding of an epinephrine, which is an adrenaline molecule secreted from the adrenal gland at the top of the kidney in response to stress, goes into the bloodstream, bounces all over the body, and by sheer luck, at some point, it's going to lock into a receptor like a key into a lock, and then it's going to cause this big blue box in the middle, this whole transduction pathway. And if you notice in the parentheses there, there's these 10 to the factor of 2, 4, and so on, 5. And the point is that it's amplifying the signal from one receptor bound. We're getting maybe tens of thousands, hundreds of thousands of molecules being created. And then way down there at the bottom, 10 to the 6 molecules of glucose are being created in order to be released to give the organism more energy to contract its muscles and either fight, freeze, or flight. Essentially, it's the fight or flight response to some sort of stressor. Which started this. But the point is that it's a crazy amplification of a signal. That's why it can't go on forever. You got to be able to shut this down, or else if people are having all this glucose being pulled out of their liver and muscles all the time, they're going to die. It just can't happen.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
Anyone feel like this right now? We're almost done. Here's the last thing. Apoptosis, controlled intentional cell death. When would you ever want to kill a cell? It seems so complicated and wondrous to have even one cell Why would we ever want to kill it? Well, sometimes in the development of organisms, like think of a tadpole's tail that gets, all those cells get killed, and then you have a frog without a tail. Or humans have webbing between their fingers, probably a vestige of our evolutionary past when we are in embryonic development, and those cells, I guess, deteriorate back in most people, and so you just have a tiny little piece of webbing at the bottom. But anyway, those are some reasons why. Because I guess we used to swim more. We don't need that any longer. Those are some examples of when cells die intentionally. That's fine. Other times, cells get signals to kill themselves intentionally, and that may occur if they have been infected by a virus, or there's just a lot of trouble. Something's going wrong with the cellular machinery, and what I mean by that is, like, most often the machinery that helps the cell divide, if something senses, and I'm not trying to be vague, most often it's the immune system that senses that something's wrong in a cell, it will stimulate the cell to kill itself. And that's better than having a cell reproduce millions of times that is bad, and then you can essentially get potentially a tumor or something like that. This is apoptosis, programmed cell death, integrates multiple cell signaling pathways. It's programmed or controlled cell suicide. Here's the example I gave you. It's essential in the development of many organisms. See that webbing. Way down there in the bottom left-hand corner, I want you to find the word apoptosis. And I want you to find at the bottom where it says death factors. You have in every cell receptors that will be able to bind something called a death factor. I'm about to show you this in a movie. And then if you follow the arrows there up from death factors, I want you to find caspase 8, and above that you see caspase 9. You're about to see the words caspase again. These things directly cause the cell. Basically what happens is the infrastructure, the superstructure holding open the cell crumbles, and then the cell implodes on itself. I want you to be amazed here. I want you to clear your mind, put everything away, and just be amazed. It's only 4 or 5 minutes long. This is happening in your body right now, probably 1,000 times at this moment. Here it is. just like magic every time I see it. Be overwhelmed by what's occurring in every one of your cells and make sure to never touch a doorknob again. You want to keep every cell possible. In fact, I would say don't wear socks again ever because you don't want any skin cells coming off in your socks. We're done with chapter 11. Be amazed, be in awe of how wonderful